Hi, my name is Jacob Gardner, and today I will be talking about flow cytometry. Now you might be wondering what exactly flow cytometry is. Well, in cell biology, flow cytometry is a laser-based biophysical technology that enables a way of identifying and counting different types of cells, and it also allows you to do this with proteins as well. And kind of the way this works is cells are suspended in a fluid and they're passed through a laser-based electron detection system, and that kind of spits out exactly what the identity of the cells are and some of the properties it has. It also allows for the analysis of both physical and chemical properties of a certain cell. So you might be wondering what exactly flow cytometry machine looks like, and this is a picture of what you'd find in maybe a cell biology lab, but you'd probably find them more in hospital labs or in clinics. And this next slide is a picture of exactly kind of what's going on in flow cytometry. So like I said before, these right here, it's a fluid suspending the cells, and this is kind of like a funnel, and what it's doing is it's funneling the cells down into where there's just one single cell going through the laser at a time. And what this does is it allows this machine to be very precise and very accurate in the measurements that it has. And so what happens is this laser goes through the cells right here, and it produces light that goes off, and it's detected by these detectors. And so it will either produce forward scatter or side scatter. And you might be wondering what forward scatter is, but forward scatter, it's kind of like an eclipse. Whenever the moon goes by and the sun shines on it, it kind of spreads the light out. And that's what it's doing right here. Forward scatter, the light's getting spread out. And then side scatter, in different cells, you have different properties in them, you have different things, and it's oriented differently. So it might reflect off and go sideways. And right here you have the different wavelengths are detected by different sensors. And these detectors are hooked up to a computer which analyzes the data that gets put to it, and then it kind of gives you the identity and the different properties of this cell. Okay, so you might be wondering how exactly is this practical in real life? Okay, so as you might know, cancer is a very big deal in our society, and 1 in every 4 deaths in America are due to cancer. And so you might really want to know when you have cancer so you can get treated for it, and flow cytometry is a good way to identify cancer cells. It also allows you to identify specific types of different cells, and kind of the way it does that is whenever you have cells that are suspended in a fluid in a sample, you treat those with antibodies, and if those antibodies attach to the cell, then whenever they're passed through the laser, it'll detect light, and that's sensed by the flow cytometry machine. Go back. Sorry. Okay, and then another way that you can detect them is through DNA. The DNA gets stained, and whenever the DNA is passed through that, it shows you how much DNA is in there. And if you have a very high amount of DNA, then that could be evidence that you might have reoccurring cancer or something like that, and you can get that treated so that it might not ever happen in the first place. Okay, so today I kind of told you all what flow cytometry is and how it identifies specific cells and their properties, and then I also showed you all how it was practical in the medical field to identifying cancer and different types of cancer. So I hope you all have a better understanding of flow cytometry after I said this, and thank you.